allow myself to read the semblance about you in Spanish and in English. La doctora Dawn Holmes obtuvo su doctorado en microbiología en la Universidad de Massachusetts Amherst. Se licenció en biología en la Universidad de California, Santa Cruz. Actualmente es profesora en el Departamento de Ciencias Físicas y Biológicas, Western New England University. Realizó colaboraciones con el Instituto Conjunto del Genoma del Departamento de Energía de los Estados Unidos, Centro de Investigación de Bosques Tropicales Australia, Servicio Biológico Nacional en Kauai, Servicio Biológico Nacional en Hanaui, etc. Ha sido profesora visitante en el Laboratorio Nacional de Ciencias de los Materiales de Shenyang en la Universidad del Noreste China y en la Facultad de Ciencias Ambientales e Ingeniería, Universidad Forestal de Beijing. Es autora o coautora de más de 200 publicaciones, tiene un índice H de 52, un índice IRIES de 87, cuenta con 12.912 citas en total y tiene un total de publicaciones revisadas por pares de 104. Dr. Dawn Holmes received her PhD in microbiology from the University of Massachusetts Amherst and her BS in biology from the University of California, Santa Cruz. She is currently a professor in the Department of Physical and Biological Sciences, Western New England University. She made collaborations with the U.S. Department of Energy, Joint Genome Institute, Tropical Forest Research Center, Australia, National Biological Service on Kauai, National Biological Service in Hanaui, etc. She has been a visiting professor at the National Material Science Laboratory in Shenyang, Northeast University, China, and the School of Environmental Science and Engineering, Beijing Forestry University. She has authored and co-authored more than 200 publications, has an H index of 52, I-10 index of 87, and 12,912 citations in total, and has a total peer-reviewed publications of 104. Dr. Holmes, it is a pleasure to have you as a speaker today. We thank you profusely for accepting our invitation, and without any further ado, we yield the floor to you.
Okay, thank you. Okay, so I'll share my screen now. Okay. Okay, so my talk, it focuses on diet or direct interspecies electron transfer between a iron-reducing bacterium called Geobacter metallireducens and different electron-accepting partners. So a lot of my research focuses on methane production by methanogens. So methanogens are archaea, and they're the only organisms that are known to be able to convert carbon dioxide, so they can reduce carbon dioxide into methane using different electron sources, things like hydrogen gas, acetate, methanol, and formate. Methanogens are abundant in anoxic environments because they cannot grow in the presence of oxygen. And you find them in places like in sewage, deep surface soils and sediments, and also the intestinal tracts of different types of animals, like a cow rumen. So methanogens, one feature of methanogens is that they cannot use complex organic compounds. They're limited to C1 compounds, so C1 meaning just one carbon, and they can use compounds like hydrogen, CO2, formate, methylamine, and methanol. So that's what most methanogens are limited to. And then there's some methanogens, which is a smaller subgroup, that can use C2 and C3 compounds like acetate or trimethylamine. So these methanogens are further classified by the substrates that they can use to produce methane or for methanogenesis. So you have three main groups of methanogens: the hydrogenotrophic methanogens, and these are the ones that use hydrogen CO2. You have the acetoclastic methanogens, which is a very small group of methanogens, and those are the ones that we mostly focus on. And they can use acetate. Or you have the methyllotrophic methanogens, and they can use methylamines or trimethylamines as sources of carbon and energy. So these different types of methanogens use different pathways, different metabolic pathways to make methane. I mean, the end steps of the pathway are shared by all of the methanogens, these last two steps. But the steps before they differ depending on if it's a hydrogenotrophic methanogen, an acetoclastic methanogen, or a methyllotrophic methanogen. So three different pathways. So if a methanogen can use hydrogen as its source of electrons, it will use the CO2 reduction pathway. If it uses acetate, if it metabolizes acetate or uses acetate as its source of electrons, it uses the acetoclastic pathway. Okay. So the role of methanogens in the food chain, in the different environments where you find it living, is usually down at the bottom of the food chain. In places like bioreactors, you have, or in sediments, you have complex organic matter that is broken down by a variety of microbes like fermentative bacteria. And then they produce the simple compounds like acetate, hydrogen, formate, and then the methanogens can use those compounds and make methane. So usually they're way down at the bottom of the food chain. So most methanogens, or the ones that we're studying, can form syntrophic partnerships with some of these bacteria, and this permits degradation of some compounds that would not normally be thermodynamically favorable. And so this is a syntrophic partnership, and our lab has been studying a variety of syntrophic partnerships between an electron donating organism and an electron accepting organism. So the methanogen is the electron accepting organism. So one type of syntrophy that is observed in nature is called indirect interspecies electron transfer. And that's when an organism, like a bacterium, for example, Desulfovibrio vulgaris, it produces hydrogen as a fermentative byproduct. And then the methanogen, if it's a hydrogenotrophic methanogen, can take that hydrogen and reduce carbon dioxide to methane. So this is an indirect electron transfer because the two cells do not need to make direct contact with each other. Another type of interspecies electron transfer, which is what we're focusing on in these studies, is called direct interspecies electron transfer. And in this type of transfer, the two cells need to make direct contact with each other, and electrons are directly transferred from the electron donating bacterium to the electron accepting bacterium, and CO2 is reduced to methane. So in this picture, this is showing electron transfer through conductive filaments. In some cases, conductive filaments that are made by the electron donating bacterium make contact with the electron accepting microbe, and then the electrons are transferred. In other cases, the two cells need to be directly attached to transfer electrons, and they do not rely on the conductive filaments. In this other type of interspecies electron transfer, humic related compounds like AQDS or humics, they serve as the intermediary or the electron donor for the electron accepting partner. So the first organism, the electron donating partner, it transfers electrons to humics or AQDS, which is a humics analog, and it reduces AQDS to AHQDS. And then those electrons, the reduced AHQDS, can then be transferred to the electron accepting partner. So this is called quinone-mediated interspecies electron transfer. It's also called quiet. So this is another mechanism, and you would see this mechanism in subsurface soils where you find a high concentration of humic compounds. And then finally, the last type of interspecies electron transfer, it is technically diet because there's only two cells that are in direct interactions with each other, but there is a conductive compound in between them. So in some cases, this conductive compound is activated charcoal, granular activated charcoal, or GAC, or it could be carbon cloth, or any sort of conductive material. So in this example, Geobacter, it can oxidize an organic compound, transfer those electrons to a conductive material, and then the methanogen, which in this example is Methanosarcina barkeri, can accept those electrons directly from the conductive surface. So they can use the conductive surface as a way to transfer electrons. And sometimes this helps diet work a little bit faster, because the two cells don't need to be in direct contact with each other. They just need to attach to a conductive surface. Okay, so in order to facilitate diet, or in order to study diet co-cultures, it takes a pretty long time. On average, at least 30 days. It's easier to form co-cultures between two Geobacter species than it is to form co-cultures between a Geobacter and a methanogen, but it takes a minimum of 30 days. So you have to be very patient, and you have to wait for aggregates, these little red balls. So the red color is the cytochromes in the Geobacter. So Geobacter are naturally red. And so they form these balls, these clusters, with the methanogen, and then they're in direct contact with each other, and they can transfer electrons, to each other. So usually the electron donating bacterium in diet, if you want it to work, you want to use a bacterium that can transfer electrons out of its outer membrane or out of its cytoplasmic membrane. So for the most part, or all the studies that have been done so far, you need to use an exoelectrogen. These are metal respiring bacteria that can get electrons out of their cell naturally because they need to reduce extracellular electron acceptors like insoluble iron oxides. So the electron donating bacterium needs to have that ability because your electron accepting organism is just like an insoluble acceptor, right? It needs to get electrons that are pushed out of the other cell. In order to ensure that the electron accepting partner is not just using hydrogen for growth, like any hydrogen that might be generated by the electron donating bacterium, ideally, you want to use an electron donating partner that does not generate hydrogen, like Geobacter metallireducens. And then also, ideally, you'd also want to use an electron accepting microbe that cannot use hydrogen. So it just makes it a lot easier to study diet when you can eliminate hydrogen from the equation. So some of our first studies of diet were between Geobacter metallireducens and different species of methanogens like Methanosarcina or Methanothrix. So when Geobacter is grown alone, without an electron acceptor and with only ethanol, it cannot grow. Okay, so Geobacter with just ethanol cannot grow because it cannot reduce carbon dioxide, and carbon dioxide is the only electron acceptor in the medium. Methanosarcina and Methanothrix species cannot oxidize ethanol. So they would not be able to grow alone. Even though they can reduce carbon dioxide, there's no donor for them to grow. So alone, neither of these species could carry out this metabolism, but together they can. So most of our diet co-culture studies are done using ethanol as the electron donor and CO2 as the electron acceptor. Okay, and so when Geobacter metallireducens oxidizes ethanol, it does produce a little bit of acetate at the very beginning. So at the very beginning, the methanogen can use some of that acetate, but it doesn't account for all of the electrons that are used to reduce the carbon dioxide. So a little bit of acetate is formed at the very beginning, but then it's rapidly depleted. And then all of the electrons for CO2 reduction are generated by direct interspecies electron transfer. Okay, so when a hydrogenotrophic methanogen grows by indirect interspecies electron transfer, it relies on hydrogenase proteins found in its membrane and then so first hydrogenase proteins oxidize the hydrogen gas and then transfer those electrons to an electron transport chain that then reduces carbon dioxide to methane. If during diet, if the electron accepting partner cannot utilize hydrogen, it does not have these hydrogenase proteins. So what happens is the electrons are directly transferred somehow to the electron transport chain to reduce carbon dioxide to methane. So Methanosarcina and Methanothrix are some of the only genera of methanogens that grow by acetoclastic methanogenesis. So that means that they grow with acetate as their donor for methanogenesis. Although they have the acetoclastic pathway, and that's usually the pathway they use, they also have genes coding for the CO2 reduction pathway. Remember, this is the pathway for hydrogenotrophic methanogenesis. So they also have all of these genes. And normally when the methanogens are growing in the presence of acetate, they do not use these enzymes, okay? Because they don't need to. They use the enzymes required for acetoclastic methanogenesis. But we found that when the cells are growing by diet, then all the methanogens that we've studied, these are diet co-cultures with Geobacter metallireducens as the electron donor, and 4 different species of methanogens, Methanosarcina barkeri, Methanosarcina acetovorans, Methanosarcina subterranea, and Methanothrix thermoacetophila. So we found that in all of these cases, when cells were grown by diet, then the CO2 reduction genes were being highly transcribed. And so basically they're using a different metabolic pathway when they're grown by diet than when they're grown by acetate dismutation. So diet, we've found, is abundant in a variety of environments. So environments that are perfect for diet, first of all, they need to be anaerobic because both the metal respiring organisms and the methanogens are anaerobic microbes. You also need to make sure that it's depleted of other electron acceptors besides carbon dioxide. So usually cells will prefer to use other acceptors over forming a diet partnership. So if the environment's rich in iron or rich in sulfate, then it might not form these diet aggregates. So you want to have it depleted of most other electron acceptors so that really the only option is for them to form a partnership with carbon dioxide as the acceptor. Another feature, or just in the environments that we've studied, is that diet occurs more frequently in environments that are rich in species of Geobacter and also species of Methanosarcina, the methanogens from the genus Methanosarcina, or the genus Methanothrix. Both of these are acetate or acetotrophic methanogens. They both use acetate as they can both grow with acetate to make methane. So first we looked at flooded rice paddy sediments to determine whether or not diet might be occurring in this environment. So we made some RNA-seq libraries and then looked at the sequence results from these RNA-seq libraries. And first, what we found is that they were dominated by Geobacter. Okay, so many Geobacter species, also many other metal respiring bacteria that could theoretically also be capable of diet. So good electron donating bacteria. And then we also found good electron accepting methanogens. So there was a huge population of Methanosarcina species and also Methanothrix species. So that's what we first determined, was that the proper organisms were present in this environment. And then what we did is we looked at those same RNA-seq libraries to see which genes were being most highly transcribed, because we had already found that in pure culture studies, CO2 reduction genes were highly expressed. And we found the same case when we looked at the flooded rice paddy sediments. So many of the genes in Methanothrix were being, for the CO2 reduction pathway, were being highly transcribed. And Methanothrix species can only use acetate or grow by diet to make methane. So normally they only use the acetoclastic pathway. So the fact that they were using the CO2 reduction pathway indicated that they were definitely growing by diet. We looked at another community, an artificial community. These were anaerobic digesters that were treating brewery waste. And this also showed, first of all, that Geobacter and Methanothrix. So Methanosata is the same as Methanothrix. Recently, the genus, some people call it Methanosata, some people call it Methanothrix. Okay, so we found that Geobacter and Methana. So am I muted? Okay.
No, we can hear you good.
Oh, okay. It said on my screen that I was muted. Okay, so anyways, so abundant in Geobacter and Methanothrix species. And then we also found, just like in the rice paddy sediments, that genes from the CO2 reduction pathway were being highly transcribed by Methanothrix species. So in these initial studies, we showed that Methanosarcina and Methanothrix use the CO2 reduction pathway for methane production during diet. So that we figured out, but we still had two important questions. So first, how are the electrons getting transported out of the exoelectrogen, out of the electron donating partner? And then how are those electrons, once they get out, how are they being taken up by the methanogen, or the electron accepting partner? So we have shown diet in two general types of electron accepting partners. So we have some that have outer membrane C-type cytochromes on their surface. So they are outer surface, I'd say, because Methanosarcina does not have an outer membrane. So outer surface C-type cytochromes. And those would include species like Geobacter sulfur reducens, which can serve as an accepting partner, or these two Methanosarcina species, Methanosarcina acetivorans and Methanosarcina subterranea. So that's one group of accepting partners that we've studied. And then the other group of accepting partners that have been studied are Methanosarcina barkeri, which does not have any surface C-type cytochromes, Methanothrix thermoacetophila, also no surface cytochromes, and same with Methanobacterium species. So that means the surface of the cell is pretty different. Okay. So when we looked further into the different groups of methanosarcina, which are excellent electron accepting partners, we further found that even within the genus Methanosarcina, you have two different groups. So type 1 Methanosarcina, which are all the ones highlighted in red, these species do not have multi-heme outer surface C-type cytochromes. They also, all of these species can use hydrogen as a source of electrons in addition to acetate. So all the Methanosarcina can, or most of them can use both. All of the red ones can use hydrogen, and most of them can also use acetate. The ones in blue cannot use hydrogen, and they have multi-heme C-type cytochromes. So they can only grow using acetate or methylamines. So when we looked further into the Methanosarcina in different, that were isolated from different environments, we were able to group them into two clusters. As I mentioned, not only phylogenetically do they cluster differently, but they're also found in different types of environments. So the type 1 Methanosarcina, which are highlighted in yellow, these are the ones that are able to grow by hydrogenotrophic methanogenesis, okay? So this means that they have hydrogenase proteins embedded in their membrane, specifically the ECH hydrogenase. And they do not have what's called an RNF complex, which is another electron transport complex in the membrane. And they do not have surface-associated multi-heme C-type cytochromes. The type 2 Methanosarcina, on the other hand, they have more than one multi-heme surface-associated C-type cytochrome. They have RNF complexes, or the RNF complex, which is involved in so it's an electron transport chain. And they cannot use hydrogen. So very different metabolisms. And they're found in very different types of environments. So the ones that can use hydrogen, the type 1 Methanosarcina, are usually found in environments that are very rich in organic matter, and also where you'd find a lot of hydrogen that the organisms could use. So a lot of anaerobic digesters are full of type 1 Methanosarcina. Type 2 Methanosarcina, on the other hand, they're found in nutrient limiting environments, places like groundwater from the subsurface or sediments from a marine canyon. So environments where you are not likely to find a lot of hydrogen, and they need to eke out their living on, few nutrients. So when we did RNA-seq libraries, we generated RNA-seq libraries from different co-cultures. So we have 5 different co-cultures with Geobacter metallireducens as the electron donor and different electron accepting partners. And we looked at patterns of expression by Geobacter metallireducens. We found some definite patterns. So first, when we looked at this is a diagram showing similarities and differences between the Geobacter metallireducens transcriptome grown by diet with different accepting partners. So when it was grown with Geobacter sulfur reducens, which has many, many multi-heme C-type cytochromes, and many of them are in its surface. That's why it looks bright pink. We found that expression patterns by Geobacter metallireducens were very different from when it was grown with the methanogens. But what was interesting is that we also saw differences in expression patterns by Geobacter metallireducens between different types of methanogens. So both type 1 methanosarcina, and the example that we used was methanosarcina barkeri, which does not have outer membrane or outer surface cytochromes, and methanothrix, which also does not have outer surface cytochromes. The transcriptome for both of metallireducens was similar with both of these. And then two type 2 methanosarcina, which both have outer surface multi-heme C-type cytochromes, Geobacter metallireducens behaved similarly with these guys. So then we looked further at expression patterns for genes coding for outer surface or just C-type cytochrome proteins in Geobacter metallireducens. And we also found that the patterns differed depending on the electron accepting partners. So just like with that diagram that I showed in the last slide, growth with Geobacter sulfur reducens was most different. And then growth with the type 2 methanosarcina was most similar, and growth with the type 1 methanosarcina barkeri and the methanothrix was also most similar. Specifically, however, we found that an outer surface C-type cytochrome, GMET0930, which is homologous to OMCZ in Geobacter sulfur reducens. This cytochrome was important for all of the conditions, and we did genetic studies that also showed that this specific outer surface C-type cytochrome is important for diet with all partners. So then we looked at, we wanted to look at the role that conductive pili played in electron transfer during diet. So we took a strain, we established or tried to establish diet co-cultures with the strain of Geobacter metallireducens that had non-conductive pili. So these cultures could not ever establish diet with Geobacter sulfur reducens. And then we also found that after 300 days, we could not establish co-cultures with methanosarcina acetovorans, okay? So we found that pili were being highly expressed by the Geobacter, and they were helping to make contact with the acetovorans and the Geobacter sulfur reducens to establish diet. However, when we looked at co-cultures with Geobacter metallireducens and methanosarcina barkeri, we found that after 80 days, we were able to establish diet. And the cells, they had to be in very close proximity to each other, basically like a sandwich. So you have a sandwich of methanosarcina with with the geobacter right here, 2 geobacter rods, and they have to be in very direct contact with each other in order to participate in diet. So when we looked at methanothrix and Geobacter metallireducens, we found a similar pattern. We have not done genetic studies yet, but these are some electron micrographs of Geobacter sitting on the surface of a methanothrix cell, and you can see very, very direct contact. And when we look at the transcriptome of methanothrix and Geobacter metallireducens, we do not see significant differences in expression patterns for genes involved in conductive pili expression. Okay, so similar to Methanosarcina barkeri, it appears that conductive pili are not important for diet with methanothrix species. So this is our proposed model for electron transport from Geobacter metallireducens to the type 2 Methanosarcina, the ones with the C-type cytochromes, and also the Geobacter sulfur reducens. So for these organisms, it appears that pili are important for establishing diet. But in all cases, this gene or this C-type cytochrome, 0930, is also required for diet. Okay. And then slightly different pathway used to transfer electrons from Geobacter metallireducens to type 1 Methanosarcina, like Methanosarcina barkeri, and the methanothrix species. It does not appear, conductive pili do not appear to be important, but 0930 is critical for electron transfer. So that was the looked at the donating organism, the Geobacter metallireducens. Now we wanted to look at the accepting organism and figure out which mechanisms might be being used for electron uptake by the methanogenic partner. So one nice thing about Methanosarcina barkeri is that it can grow both through interspecies hydrogen transfer, because it can use hydrogen, and it can grow by direct interspecies electron transfer. So that means that you can do direct comparisons of Methanosarcina barkeri, either growing by accepting a hydrogen from a fermentative organism that produces hydrogen, or growing by diet from Geobacter metallireducens, which does not generate hydrogen. So what we found is that even though genes for the transcriptome showed that genes for hydrogenase proteins were definitely more highly expressed in cells grown by interspecies hydrogen transfer. But we were intrigued to find that some of these hydrogenase genes were still being pretty highly expressed, even when the cells were grown by diet. And that's because studies have shown that Methanosarcina barkeri uses hydrogen cycling during growth. So even when it's growing by acetate, it will produce hydrogen. And so the hydrogenase proteins are still active at all time in this methanogen, which really makes things complicated. So much easier to study diet when you don't use a hydrogenotrophic methanogen. Okay. But we also found that many of the genes coding for the FPO dehydrogenase complex were much more highly expressed in diet-grown cells. And so what it appears that normally the FPO dehydrogenase complex, it accepts electrons from a reduced F420 and it pumps protons out of the cell. But it appears that during diet, it works in the opposite direction. Protons can be pumped into the cell and F420 can be reduced to the reduced form of F420. In addition, we found several different types of surface-associated redox proteins that were more highly expressed in diet-grown cells. We still couldn't exactly figure out which one was the most likely candidate, but these were the redox proteins because there's no C-type cytochrome in these Methanosarcina barkeri, but there were some redox proteins that were more highly expressed. So our proposed mechanism for electron uptake by Methanosarcina barkeri, based on our studies, was that some sort of electron carrier accepts electrons that are transferred from the partner and then transfers those electrons to methanophenazine, and then F420 is reduced. So we found in the transcriptome of methanothrix thermoacetophila that FPO dehydrogenase was also being highly expressed in diet-grown cells. And there was a quinoprotein, which is homologous to the quinoprotein right here in Methanosarcina barkeri that was also highly expressed. Okay. And so this surface-associated quinoprotein was also being highly expressed, and it's possible that this is what accepts electrons in the membranes of methanogens without C-type cytochromes, and then transfers those electrons to FPO dehydrogenase to reduce F420, which then is a source of electrons for methanogenesis. So we also looked at Methanosarcina acetovoran, so electron uptake by this type 2 Methanosarcina. And one feature of this methanogen is that it has an RNF complex. Okay, an RNF complex is an electron transporter, and it was highly expressed during growth by diet, much more highly expressed than it was during growth with acetate. And also specifically a outer surface C-type cytochrome with 7 heme groups, MMCA, was also highly expressed. And when we looked at the transcriptome of the other type 2 Methanosarcina, we found the Methanosarcina subterranea, similar results were observed. So both the RNF complex and this one multi-heme C-type cytochrome were highly expressed during diet. So we did genetic studies with Methanosarcina acetovorans, where we deleted the gene for that C-type cytochrome, MMCA, and we found when we deleted that gene, it could not establish diet. So it definitely is required for electron uptake, this outer surface C-type cytochrome. We were also intrigued to find that when we knocked out the archaeal flagella, which is called an archaeella. When we knocked that out or deleted that from Methanosarcina acetovorans, cells also could not establish diet. So one interesting feature of archaeella from type 2 Methanosarcina, like Methanosarcina acetovorans, is that it's rich in aromatic amino acids. So a couple of features of conductive filaments that have been found is there's two important features. One of them is that they need to have at least 9% aromatic amino acids in their mature filaments. So after the signal sequence has been cleaved. And then they also need to have a gap between those aromatic amino acids that's no greater than about 50. So we found that there were the ones that we knocked out, the archaeella that we deleted, both were theoretically or putatively conductive. So based on our results from our transcriptomic and genetic studies during diet with between Geobacter metallireducens and Methanosarcina acetovorans, we found that electrons are accepted by MMCA, and then they're transferred to the RNF complex. I forgot to mention that FPO dehydrogenase was also highly expressed. So that is highly expressed by all of the methanogens. And then so both FPO dehydrogenase and the RNF complex are involved in electron uptake in the membrane of Methanosarcina acetovorans and Methanosarcina subterranea. Okay. So finally, we wanted to know whether or not addition of some sort of shuttle like riboflavin could enhance diet between Geobacter and a methanogen. So previous studies that have looked at diet between Geobacter metallireducens and Geobacter sulfurreducens have shown that riboflavin helps with diet, so it enhances diet between these two species. However, in these studies, they were using fumarate as an electron accepting partner. And so uptake from riboflavin to cells that are then using fumarate as an electron acceptor is thermodynamically favorable. When we tried adding riboflavin to co-cultures between Geobacter metallireducens and Methanosarcina barkeri, however, it did not enhance diet at all. And so it could be because electron transfer to carbon dioxide from riboflavin is not thermodynamically favorable. Okay. So in conclusion, both Methanosarcina and Methanothrix species use the CO2 reduction pathway during diet. Second, diet between exoelectrogens and Methanothrix or Methanosarcina is occurring in both natural and artificial environments. So places like flooded rice paddy sediments or wetlands and anaerobic digesters. Geobacter metallireducens uses different routes for electron transport out of the cell when it's grown in the presence of different electron accepting partners. The outer membrane 8-heme cytochrome, GMET0930, is important for electron transfer from Geobacter metallireducens toxins to all accepting partners. The FPO dehydrogenase complex and potentially quinoproteins are involved in electron uptake by methanosarsina and methanothrix thermoacetophila. The outer surface cytochrome, MMCA, and also the RNF complex are required for electron uptake by type 2 methanosarsina. And then finally, riboflavin does not enhance diet between Geobacter metallireducens and methanosarsina barkeri. Okay, so that's it. Thank you.
Thank you very much for your interesting presentation, Dr. Holmes. We will now give the floor to some participants to express doubts and comments. Dr. Carlos Escamilla, would you like to make your question?
Yes, thank you, Mariana. Thank you, Dr. Holmes. Nice to meet you, finally. Only by email we were having contact.
Yes.
Well, I appreciate, I thank for your presentation, which is very clear, even for people that are not expert in such particular topics as genetics and enzymes and all this diet metabolism. And also because of the way you spoke English, it's very understandable and clear for non-native English speakers.
Oh, good. Thank you.
Yeah, it's very nice to of your part. I have many questions, but
Okay.
I will have time for mine only. I will only like to know which one. What are I know that the work that you shown us is it's a lot of work. It's a lot of laboratory hypotheses and tests. But what would practical researchers, as me, I work with anaerobic digestion and hydrogen production. What can we, as practical researchers, learn from your work and from all this diet metabolism?
Okay. So we've shown, especially in anaerobic digesters, that when you enhance diet, because basically it allows you, because I showed that food chain, right, where the food chain with methanogens at the bottom, right? And that's a pretty complex food chain that can take a long time. So if you facilitate diet in digesters by adding things like conductive materials, it increases methane production and degradation enormously. So if you want, especially if you want to degrade compounds rapidly, it happens much faster if you stimulate diet. Because basically you're removing all of those intermediates, right? And then you can break down complex organic matter, more complex like propionates, okay? And then convert it to methane and then use that methane as a biogas.
Yes, like accelerate the anaerobic digestion, which we know it's rather slow compared to other reactions, right?
Yes, yes. And also when you accelerate the anaerobic digestion with diet, then you get fewer, volatile fatty acids accumulating, and the reactor is healthier. It's less likely to crash, right? Because fewer acids are produced. Sorry, my light just, let me just, okay.
Yes.
Sorry. Okay, so yeah, so your reactor is healthier also if you stimulate diet.
Yeah, and that may lead to new reactor designs and all that stuff, right?
Oh, yes, yes, yeah, it makes a really big difference. I have a few papers that are out that show how much better the reactor performs when diet is stimulated.
Fine. Well, I will pick a question from the audience. I will pick the last one, which is by Dr. Katy Juarez. What happens? She says, thanks for the excellent talk. What happens in nature parasites when there are many organisms with different metabolism? It is known who establishes the preference of a certain syntrophic relationship. For example, if the Sulfovibrio, Geobacter, and different species of Archaea coexist.
Oof, that's complicated.
Yeah.
I mean, that is research that we're starting to look into. I think it really depends on the environment, right? And it depends on, I don't know if I can exactly answer the question, who would win? I think Geobacter is definitely better at establishing diet than Desulfovibrio. That's what I found. And so Geobacter are more likely, and I don't know because I haven't done a lot of work with methanogens other than Methanosarcina and Methanothrix because they're clean, right? They're easy. But when when a methanogen can use hydrogen, it just makes everything so complicated. But if hydrogen isn't available in the environment, then probably the only way they could make methane would be to establish diet. So environments with just carbon dioxide and not many other acceptors are going to be richer in diet. I don't know if that answered the question. But
well, I think that would be a very extensive answer for exactly pointing out every kind of syntrophic relationship.
Exactly, because I think it would vary depending on the environment, right? Like environments are so complicated.
Yeah, well, Dr. Holmes, I give the word to Dr. Giovanni because we are now on time, and at 12 o'clock, it's the individual sessions. So, Giovanni, your turn. Thank you, doctor, for your answer.
You.
There are many